Start, madam.
Good morning, students. Welcome back to another session of plant physiology. Chapter number 4, photosynthesis in higher plants. We have started this chapter and you learned about the historical background of scientists for the understanding of photosynthesis. And in the second class, you have learned about site of photosynthesis, that is chloroplast, the structure of chloroplast, structure of chlorophyll pigment molecule, and the types of chlorophyll molecules also you are familiar with. In the third session, we have started the light reaction, reaction center, light reaction, and photosystems. Photosystem 1, photosystem 2, cyclic photophosphorylation, and non-cyclic photophosphorylation. But as time was not sufficient, we were unable to finish that slide. So today, the same slide I am going to continue. And then afterwards proceed with the comparison of the photosystems as well as the cyclic and non-cyclic photophosphorylation. So let us start with this light reaction. What is light reaction? As you all know, the reaction that takes place in the presence of sunlight, that is photochemical phase. It includes light absorption, water splitting. Water molecule is going to split and oxygen is released. And when the water is splitting, the hydrogen protons and electrons are also getting separated. And these are taken by electron acceptor molecules. And the most important formation of high chemical intermediates, ATP and NADPH. NADP is a electron acceptor molecule. It is going to accept the protons from water molecule and electrons released by the chlorophyll molecule. And in this light reaction, photolysis is the term which was introduced for you. That means splitting of water molecule, releasing protons, electrons, and oxygen. And photophosphorylation. Photo means light. Phosphorylation is nothing but addition of inorganic phosphate to adenosine diphosphate and making it as adenosine triphosphate, which is the energy currency of any cell. So this ATP is synthesized here in the chloroplast in the presence of sunlight. That's why it is called photophosphorylation. And it is of two types, non-cyclic and cyclic. Non-cyclic, because of the movement of electrons, it is called as non-cyclic. And if the electrons are revolving in a circular path, it is called cyclic photophosphorylation. These two things we are going to discuss. Reaction center. For any chemical reaction to take place, a reaction center will be there. First of all, quantazome. Quantazome is nothing but these chlorophyll pigment molecules which are present in the thylakoid. So this is a thylakoid, and in this chlorophyll pigment molecules are present which are going to make antenna molecule. These are antenna molecule because they are going to trap the solar energy. You can see the chlorophyll B, carotenoids. These are the accessory pigments. They are the accessory pigments. They are the helping pigments. They will also trap the solar energy. After trapping this solar energy, these antenna molecules are going to give it to the chlorophyll A, which is going to make the reaction center. And this complete setup is called light harvesting complex. So a light harvesting complex is there. And then in this light harvesting complex also, you'll see photosystem 1, photosystem 2. This photosystem 1 and photosystem 2 is nothing but it is the light harvesting complex only, occurring at particular wavelength of light, that is 680 nanometer wavelength and 700 nanometer wavelength. 700 nanometer wavelength is photosystem 1. 680 nanometer is photosystem 2. Okay, so these two important points you have to remember, PS1, PS2, or we can also say P700, P680. Depending on the wavelength, we are going to give the names. So here, properly, everything is going to proceed in this light harvesting complex. Chlorophyll A is the boss, which is going to trap the entire energy, collect all the energy from the antenna molecules. And here, the chlorophyll A molecule electron is going to jump. Jump because now from low energy state, it has attained high energy state when it is getting the energy. A pair of electrons are going to jump from the reaction center. As you can see clearly, the photons are nothing but the light travels in small packets called photons. Photons will be having the quantum energy. This is taken by the pigment molecules, where you are having a reaction center chlorophyll A molecule. Reaction center chlorophyll A molecules, a pair of electrons are going to jump. When it jumps, the primary electron acceptor will be there accepting that electron. So this is going to be the light harvesting complex. It is common for both PS1 and PS2. This functioning is common for PS1, PS2. In the PS1 also the same, light is taken, becoming high energy state, chlorophyll A molecule electron will jump and accepted by primary electron acceptor molecule here. So P700 as well as P680. That means photosystem 1 and photosystem 2 will be having this type of setup taking place. And this is called light harvesting complex. I hope you are having a clear idea how the chlorophyll A is taking the energy and electrons from chlorophyll A is jumping. Now, what happens further we are going to learn, and here also diagrammatic manner, the sunlight energy traveling and these antenna molecules, and this yellow color is the chlorophyll A reaction center. Electron is jumping here like this. Okay, and here you can see center of reaction. Here you can see P700 and P680. At both these P700 and P680 nanometer of red light wavelength only it is taking place. Next, photosystem 2. We have started this one, and here just you can see P680 is nothing but PS2. In photosystem 2, always remember P680 wavelength. It will start. And here, when the electron is jumping, after jumping, the electron is not remaining like that only. It is going downward, downhill. Robert Hill discovered this one. And downhill, you can see how on the redox potential scale, it is losing the energy and it is just coming down. And where it is reaching at PS1, P700. So from PS2, it is going to reach PS1, the electron. Meanwhile, what is happening to that electron? The electron is not utilized. Only the energy in the electron is utilized for ATP synthesis here. And you can see PQ cytochromes, PC. PQ is plastoquinone, CTY is cytochrome, PC is plastocyanin. These molecules are passing it, and they are the electron carriers. They are the electron carriers. So this is nothing but electron transport system. So you will find electron transport system here, and the electron is moving and reaching the PS1, P700. So this is about PS2. In detail, if we'll see what is actually happening in the non-cyclic photophosphorylation. The same PS2 is here and PS1 is here. Here the PS2, the electron is jumping and it is moving from plastoquinone. It is moving from plastoquinone to cytochrome complex, then plastocyanin, and it is reaching the PS1. PS1 is 700 nanometer red wavelength. Remember that. Okay. After reaching here, this chlorophyll molecule is going to accept, because here at P700 also the same thing is happening. Light is also taken by this reaction center, light harvesting complex. Here also the chlorophyll electron has done the same thing, jumping up due to high energy state. Okay, and here also that electron is moving to ferridoxin here, and ferridoxin is transporting it to NADP, electron acceptor, and it is taking here. So here our PS1's electron is moving and coming to PS2. PS2's electron is moving and going to NADP. This thing you have to remember with clarity. So don't forget how the electron from PS2 is reaching to PS1. PS1 electron is reaching to the NADP. So this is one electron removal and accepting process you have seen. Meanwhile, when the electron is moving, here ATP synthesis is taking place. Here ATP synthesis is taking place at this point. And you can see the NADP is also accepting the electron, and it is in the presence of enzyme NADP reductase enzyme, it is getting converted to NADPH. That means it is becoming reduced. How it will become reduced? By accepting electrons and protons. You can see this H, where from this H has come here. Electrons, we know that it has come from PS1, electron is going like this and coming here. This proton has come from the water molecule. You can see here at the site of PS2, we also have a water complex here, photolysis complex. Photolysis complex. In photolysis complex, what is happening? The energy present in the chlorophyll will be utilized, and here water is going to split into protons, electrons, and oxygen. Remember, protons, electrons, and oxygen. Oxygen is the gas, it is a byproduct, it is released. What about these protons? These protons are going here and meeting with the NADP molecule and making it get reduced. And what about the electrons of water molecule? These electron molecules are taken by PS2. Remember that PS2 electrons are given off like this when it is reaching high energy state. So it wants its electron back. So it is going to take the electrons from water molecule. All this process is taking place in the presence of sunlight. And here you can see ATP synthesis, NADP, H, and also the oxygen. These three are the end products of light reaction. And reaction center is PS2, PS1. And this non-cyclic photophosphorylation is taking in the thylakoid membrane. Thylakoid membrane, the reaction center PS2, PS1 is present. And also on the membrane itself, water breaking photolysis complex is also present. So this is non-cyclic photophosphorylation. Non-cyclic because here the electron from PS2 is given to PS1. From PS1, electron is given to NADP. And this water molecules electrons are taken by PS2. So in this manner, the electron is not coming back. It is going in one direction only, and that too, you can see the shape. Z shape you can see on the redox potential scale. If we see, this one is jumping up and downhill, it is coming down, the electron, and again the electron is jumping high because of high energy. Again on the redox scale, the electron is coming downhill and the ferredoxin is taking it and passing it to NADP to make it reduced, NADPH. So this is Z scheme also. This is also called as Z scheme, a non-cyclic photophosphorylation that take place in the thylakoid membrane. Why I am particularly mentioning thylakoid membrane means this non-cyclic photophosphorylation will take place in the thylakoid membrane. What about the cyclic photophosphorylation? That will take place on the stroma lamellae. So the same electron transport points are given here, and I have taken in the previous class also how the P680 PS2 electron jumping and transporting to PS1. And in the next, you can see here how the ATP synthesis is taking place here when the electrons are moving from PS2 to PS, and from PS1 the electron is moving to the NADP. At the same time, here also the electron is carried to the NADP and it is forming NADP. And here photolysis is also taking place. Three things you have to remember: photolysis of water, ATP synthesis, NADPH synthesis. All these three are the end products here. Oxygen end product, ATP end product, NADPH. So that is all about electron transport taking place with the help of the carrier molecules here: plastoquinone, cytochrome, plastocyanin, and here ferredoxin. So you have to remember this electron transport is taking place. And finally, we will have takes place in thylakoid membrane. Photolysis of water is taking place. Yes, NADP gets reduced to NADPH2 here, and ATP synthesis take place, and that is what happening here. So this is all about non-cyclic photophosphorylation. Now we'll move on to cyclic photophosphorylation. This is only called the Z scheme, which we have completed just now. Now coming to the PS1, photosystem 1. Photosystem 1, this is present in the stroma lamellae. This is also present in the thylakoid membrane, just now you have seen. This is also present in the stroma lamellae, and it will be undertaking cyclic photophosphorylation. Why cyclic photophosphorylation? Means you can see here. In the same sunlight, photons energy is trapped by the light harvesting complex. Light harvesting complex here. The reaction center P700 nanometer red wavelength. This is the site photosystem 1, where the light is harvested and the electron is jumping and primary electron acceptor is taken and getting transported with the help of which carrier? Ferridoxin, and then to the plastoquinone, then cytochrome complex, then it will reach plastocyanin, and finally it will get back to the same chlorophyll molecule. So it is coming back, the electron which is jumping after losing the energy, getting back to the same chlorophyll pigment molecule. So this is called cyclic. And here cyclic photophosphorylation. What is the photophosphorylation? This formation of ATP. You can see here ATP is getting formed because of the energy present in the electron which is moving, and here also one more ATP synthesis is taking place. So this is called cyclic photophosphorylation, and here you can notice that there is no water complex. Splitting of water molecule releasing oxygen is not taking place at all, and NADPH is not getting reduced here. Only ATP synthesis taking place in the stroma lamellae. So this is cyclic photophosphorylation, and you have to remember where it takes place in the photosystem 1 only. Photosystem 1, which is located on the gran or stroma, will be their question. You have to write stroma lamellae. It is not neither thylakoid membrane nor the stroma. It is the stroma lamellae. I hope you remember the structure of the chloroplast. Okay, this is the stroma. Okay, and in the stroma you are having the granum thylakoid. These granum thylakoids are attached with a stroma lamellae. This stroma lamellae, this is the site for photosystem 1's cyclic photophosphorylation. Otherwise, the PS2 and PS1 located on the grana thylakoid is the site for non-cyclic photophosphorylation or Z scheme. So I hope you are following this one. PEA stands here. PEA we will write generally primary electron acceptor. FD is the ferridoxin, and PQ is the plastoquinone. CY cytochrome, and PC plastocyanin. And these are the electron transport carriers. They are transporting. They are not using the electrons for themselves. They are just passing it back to the chlorophyll A molecule in the cyclic photophosphorylation, and it occurs only in PS1. Cyclic photophosphorylation will be occurring only in photosystem PS1. And no photolysis of water. Here there is no oxygen formation taking place, no water splitting taking place, and only ATP synthesis is taking place. That you are seeing here, ATP is getting synthesized, and no reducing power, that is NADPH. It occurs in the stroma lamellae. Sorry. So next assignment 3 I haven't given that day. So note down this one. Go through the video again and again, so that if any doubt is also there, you will be learning, or else you have to revise it on your textbook pictures. In your textbook, beautiful clear pictures are there, colorful. There also you can find out how it is taking place. In the light of modern research, describe the process of electron transport, cyclic and non-cyclic photophosphorylation. This is the assignment, and you have to finish and forward it to your teachers. And this is not end of the session. Further, today's session we are having the comparison of both photosystem 1, photosystem 2, and cyclic photophosphorylation and non-cyclic photophosphorylation. This topic is somewhat huge and detailed understanding is must. For detailed understanding, you have to revise and revise so that it will be a clear vision in your mind. Once if you are reading or once if you are listening, you may understand for that time period of time. But again, when you want to recall, you'll just get stuck. So you have to revise. And here in this session of comparison, it will be very clear for you what is PS1, what is PS2 by checking out the distinctions. Before that, today I would like to congratulate, appreciate the work of Lohita and B. Harika from Vangara TSRJC and B. Harshita from Kodakandla TSRJC. Very good children. You are sending your work to me directly and I appreciate your work, the clarity in your diagrams as well as the points you are noting down during the session. Note down the points. It will be easier. If not, you are having the video uploaded. You can revise one more time during the examinations also. You can revise and have a look at the PowerPoint presentation, which is very clear cut. And here, once again, have a look at the picture. Just recall what is this picture. Just by seeing this picture, you must say it is PS1. PS1 means, as you can see here, P700, 700 nanometer red wavelength, how the sunlight photons are traveling, and at the reaction center, the chlorophyll A and the antenna molecules, the chlorophyll B, carotenoids, so these accessory pigments, these are trapping the solar energy, and the reaction center P700, that is the PS1 electron is jumping, getting excited, and it is jumping up. And here the primary electron acceptor ferridoxin is going to take that one and pass it to plastoquinone, cytochrome, plastocyanin, and finally coming back to the chlorophyll A molecule. This is nothing but a cycle, so it is called cyclic photophosphorylation. And just by looking at the diagram, you'll say that here ATP synthesis is taking place and you're not seeing any oxygen formed. That means water complex, photolysis complex is not there. And here no NADPH getting reduced, NADP is getting reduced. So here no reducing power is formed here, only ATP synthesis, and that too the cyclic movement of the electron. That's why the name cyclic photophosphorylation. So this is our PS1 and PS2, and this PS1 is located in the stroma lamellae. This point you have to remember, stroma lamellae. And by looking at this diagram also, you must recall again PS2, P680. P680, the electron is jumping and the electron transporters PQ, that means plastoquinone, cytochrome, then plastocyanin. These are helping the electron to pass and reach the PS1, P700. And here you can also see oxygen is released. That means photolysis is taking place. Water complex is also there here, where water is splitting and protons, electrons are released. Electrons are taken back by the P680 here. You can see the red arrow mark is nothing but indicating the electrons of water is reaching again this PS2. And these protons which are left over here when the splitting of water is taking place, these protons are taken by the reducing power NADP and NADPH is going to form. So you have to remember this PS2 is nothing but photosystem 2, 680 nanometer, and here the electron is given to the PS1, P700 nanometer. So let us see or compare this PS1, PS2 properly here. Reaction center PS1, photosystem 1, P700 wavelength, or simply PS1 and PS2 are alternately used here. And PS2, that is P680, 680 nanometer wavelength only PS2 will be located. And cycling, which type of cycle it is taking? In PS1, this is involved in both cyclic and non-cyclic photophosphorylation, it is present. Whereas PS2 is present only in the non-cyclic Z scheme. Next, ratio of chlorophyll and carotenoids. Here chlorophyll pigment molecules will be more, 20 to 30 chlorophyll molecules and 1 carotenoid. In PS2 also the same thing, 3 to 7 chlorophyll molecules and 1 carotenoid. And the most important characters, location, oxygen, source of electron. We'll see here location. Where is the PS1 located? The PS1 is located on the membrane of thylakoid and stroma lamellae. On the stroma lamellae only PS1 will be seen. On the thylakoid we'll see this PS1, and here also you can see PS2 is only on the membrane of thylakoid. Means on the thylakoid membrane we'll find both PS1 and PS2, but only on the stroma lamellae PS1 is located. Next, oxygen. Sorry. So here oxygen is nothing but splitting of water molecule. That means water complex must be there. Photolysis complex must be there, and you will see PS1 not associated with complex of photolysis, and no oxygen is evolved. At the PS1 you won't find any photolysis complex, and no oxygen is evolved here. Oxygen is evolved where? At the PS2, P680. P680 only. Here photolysis is taking place, so oxygen is evolved. Source of electron. Electron source is also very important. How the electrons are jumping and reaching. If any substance is losing electron, the next moment again it will gain electron. That concept you have to remember. That is called redox. Okay, reduction, oxidation. Both are going to follow each other. It is a biological process. Remember about reduction and oxidation. It is a biological process, and it is going to occur alternately, one by one. So here you will see in the cyclic, electrons come from chlorophyll A. In non-cyclic, electrons come from PS2. In cyclic, you have seen why it is cyclic, because chlorophyll A electrons only will jump, and again after passing and forming ATP synthesis come back to the chlorophyll A. Whereas in non-cyclic, from PS2, the electrons are coming from PS2. For photosystem 1, the electrons are coming from PS2. And here source of electron for PS2, it receives electrons from photolysis of water. This PS2, it is going to lose electron, and you know that electron is passed on to PS1 in the Z scheme, recon. And here the PS2 again it wants electron, means it will be taking from the water molecule which is splitting and electrons are getting separated. That electrons it is taking. So this point is very important. The PS2, the electrons are coming from the water molecule, and the PS1, the electrons are coming from PS2 in non-cyclic, and in cyclic means chlorophyll A, the same molecule only. The electron will jump and get back. That's why it is called cyclic. I hope you are following this one. Next, the comparison between the cyclic and non-cyclic photophosphorylation. You can see here cyclic. Cyclic means in a cycle form, the electron is moving. Non-cyclic means the electron is not getting back to the same position. It is just going to some other acceptor molecule, non-cyclic. And photophosphorylation means ATP synthesis will be taking place. That means for both these, ATP synthesis is common thing taking place. So here product synthesized, you can see only ATP in the cyclic. In the cyclic means in the PS1. In the non-cyclic, you will see ATP is also synthesized and ADPH2 is also formed. And byproducts. Here there is no byproduct because there is no water complex. Whereas in non-cyclic, there is a water complex. That means oxygen is liberated. Next, in the photosystem involved, Here cyclic, only PS1 is involved. Cyclic photophosphorylation means only PS1. And here non-cyclic, both PS1 and PS2 are involved. And this non-cyclic is taking place in the thylakoid membrane. And this cyclic photophosphorylation will be taking place in the stroma lamellae. You can see source of electrons. Source of electrons, here no external source of electron is required. From outside there is no need of supply of any electron, because the same chlorophyll A pigment molecule, its electron is getting back. It is jumping up high and again coming back to the same molecule. Whereas in non-cyclic, water is the external source of electron. Non-cyclic will start from PS2 and the electron from PS2 will go to PS1. From there it will be going to the NADP. From PS1 it will be going to the NADP, and here the water molecule is going to give its electron to the PS2. Okay. Next, conditions required. This is most important condition. What type of conditions it is going to work? It works in low intensity of light because it is taking place in the stroma lamellae, not the thylakoid membrane. It is taking place in the stroma lamellae, and low concentration of carbon dioxide. Okay, and also in anaerobic conditions, it will take place. Cyclic photophosphorylation. Whereas non-cyclic photophosphorylation works in optimum intensity of light and high carbon dioxide concentration and aerobic conditions. So with this we complete this session, and here some more time is there. Just you can have a recap of all this cyclic and non-cyclic just by looking at the pictures. You have to recall it. This is photosystem 1. Immediately when PS1 is, P700 must come to your mind, and electron is jumping, and electron is passing through the electron transport and getting back in the PS1, photosystem 1. Okay, this is for cyclic. This is for only and only cyclic photophosphorylation. PS1 at cyclic photophosphorylation, the electron is getting back. And by seeing the diagram, you must also identify ATP synthesis taking place. ATP is synthesized in the presence of sunlight, and the electron is going in a circular fashion. That means from the same PS1, it is jumping and getting back to the PS1 only. That means it is a cyclic, and photo means light is present, and phosphorylation means ATP synthesis taking place. So this is very simple and easy method to understand the entire concept. This diagram is enough for you to understand cyclic photophosphorylation, to know about PS1, that is P700 PS1. Okay, so this is the thing about PS1 at cyclic photophosphorylation. That means it is taking at stroma lamellae. The site is very important, stroma lamellae. Here no photolysis of water, no oxygen formation, no reducing power NADPH is formed. Only ATP synthesis, only cyclic photophosphorylation is taking place. This diagram is enough for understanding this complete concept of cyclic photophosphorylation that takes place at P700 only, where only ATP synthesis takes place. Okay, and no oxygen, no NADPH. And the site, the location is important. And remember, the location is stroma lamellae. Whenever cyclic photophosphorylation term is coming, you must remember only the stroma lamellae. Okay, at the stroma lamellae only, this PS1 setup is there. No PS2, only PS1, P700 nanometer wavelength. Next. So here next PS2. So here PS2 I am saying, but here you can see this is nothing but this is a cyclic photophosphorylation. Cyclic means, sorry, non-cyclic. Non-cyclic photophosphorylation. Non-cyclic means the electron at the photosystem 2. What is the photosystem 2? P680. The electron which is coming out, jumping up, passing through the electron transport will reach PS1, that is P700 wavelength. Because at the P700 wavelength also the same situation, the electron is jumping here also, and it is given to the NADP, passing through the FD and reaching the NADP. Okay, and here the most important thing, this PS2 680, after losing the electron, it will get back the electron from water complex, photolysis complex. You can see this H2O is splitting into oxygen, protons and electron. You can see the red arrow indicates the electron is given to P680. And you can see the H plus protons. These protons will be passed here to the NADPH. Okay, in the Z scheme you can see that diagram. And oxygen is a byproduct here. And here you can see ATP synthesis taking place when this electron is moving from this PQCTY PC, plastoquinone, cytochrome and plastocyanin. Here you will be having the ATP synthesis taking place. In the further class in detail, chemiosmotic hypothesis, you are going to learn about this. So this ATP synthesis is taking place, NADPH is also formed, and oxygen is also liberated. Where? At the photosystem 2, that is the non-cyclic photophosphorylation. The non-cyclic photophosphorylation includes PS2 and PS1 water harvesting complex. This water complex, that is photolysis complex. So this is non-cyclic. Only the thing, the electron movement is in a non-cyclic manner. From here it is moving like this, going to the PS2. From here it will be jumping, and the electron is passed to NADP. So it is non-cyclic in the Z shape. It is present, so it is called Z scheme also. And Z scheme photophosphorylation or non-cyclic photophosphorylation, where ATP synthesis will be taking place, NADPH is also formed, oxygen is also liberated, and it takes place at particular site. What is that particular site? The granum thylakoid or the grana thylakoid. The stacks of grana is called the thylakoid. So the grana thylakoid is the location you have to remember. Okay, non-cyclic means immediately PS2, PS1 water harvesting complex must come to your mind. Whenever non-cyclic photophosphorylation term is coming, you have to get this PS1, PS2, PS1 water harvesting, this water complex, photolysis complex, splitting of water and releasing oxygen. Okay, so this thing has to come to your mind when non-cyclic term is used. So have a look at this table. PS1, PS2, both cyclic and non-cyclic will be taking place at PS1 with the help of PS1. In cyclic photophosphorylation, non-cyclic photophosphorylation, PS1 is involved. But here non-cyclic only PS2 is involved. It is absent. PS2 is absent in the cyclic one. And location also, this is very important. PS1 is located on thylakoid membrane as well as the stroma lamellae. If located on thylakoid membrane, it is able to undertake non-cyclic photophosphorylation. Located on stroma lamellae, it is going to undertake cyclic photophosphorylation. And here only on the membrane of thylakoid, PS2, oxygen is not formed in the photolysis process of PS1 because photolysis complex is only not present. And here in PS2, photolysis complex is present here, from where it is going to accept the electrons also. You can see it receives electrons from photolysis of water. In cyclic, electrons come from the same molecule, chlorophyll A, In non-cyclic, electrons come from ps2. Distinction between cyclic and non-cyclic photophosphorylation. ATP is formed, oxygen not formed, occurs at ps1, cyclic. Non-cyclic, ATP and ADPH2 formed, oxygen is evolved, ps1, ps2 both are involved. In cyclic, electron source is not required because the chlorophyll A electron only jumping and coming back. And non-cyclic, from water the electron will be received by the ps2. Whereas here it works in low intensity of light, CO2 concentration low, and also in anaerobic condition. And the opposite is here, works in optimum intensity of light, high carbon dioxide concentration, and aerobic conditions. So I hope it is clear for you. Revise this again and again so that it will be very clear for you. What is cyclic, what is non-cyclic, what is photosystem, what is ps1, ps2, what is reaction center, light harvesting, and the most important in this is electron transport. How the electron is getting transported, how it is moving through the plastoquinone, cytochrome, plastocyanin, ferridoxin. So this concept, if you are clearly understood, then we can move to the next topic in the next session. I hope you will complete the assignment work and forward it to your concerned JL. Thank you. Thank you all. End of the session.